Hello students. In the last video we have discussed the application of biotechnology in the field of agriculture. So in that we have seen how to make the pest resistant plant, then how to obtain different type of other products or the different proteins from the plant, then how to enrich the nutritional quality or how to decrease the post harvest loss. So all those criterias under the agriculture we have seen. Now in this section we are going to see the use of biotechnology in the field of medicine. In this medicine also, biotechnology helps in a vast way in producing the genetically engineered insulin, then other type of products like growth hormone, then HCG, different other hormones synthetically they are produced in the biotechnological laboratories. Then after that, monoclonal antibodies where different injections, vaccinations they are made with the help of biotechnological approaches. Then after that, gene therapy, then different techniques for the just to detection for the detection of different pathogen in our body. We have different techniques. Those techniques also require biotechnology, like suppose ELISA. In this ELISA test, we have the use of biotechnology, then we can able to detect the presence of antibodies in our body, like for the disease HIV/AIDS. So these are actually the vast. We will discuss one by one. First of all, we'll see in the field of medicine. First, we are going to see genetically engineered insulin. Okay. Now see, first of all, what is the genetically engineered insulin? First of all, lot of people they are suffering from diabetes. So for this diabetes, the patients they require the insulin injection. So that insulin actually is synthesized. How actually it will be extracted? See, every individual will produce only that amount of insulin they need. They are not going to produce in excess. So how actually it is possible that this insulin they are synthesized artificially? One thing we need to know that two scientists in this approach for the first time they actually extracted the insulin from dog pancreas, and it was injected into the patients of diabetes. So what are the scientists' name? That were Benting and Best. Okay, so Benting and Best they actually collected. They just collect the insulin from the dog pancreas in 1960. Okay, in 1960 collected insulin from dog's pancreas. Okay, so later on it was seen that whenever this insulin is injected, lots of people have allergic reaction because dogs proteins they are not matching with human. Maybe the dogs have some another antigens due to what suppose it enters inside our body and our body will start to react against those specific proteins. So this is though a very easy process to collect the insulin from the other organisms, it is going to have lots of disadvantages. One of that disadvantage is the allergic reaction. Okay, so if that individual have allergic reaction, that means it is not successfully done. So that's why in 1985 for the first time genetically engineered insulin was constructed. So before coming to the genetically engineered insulin, we will see first of all how insulin actually are produced in our body. Okay, who is producing insulin? We need to know this is the pancreas. We know that pancreas is a dual organ, that means it is going to have exocrine part also, endocrine part also, right? So this pancreas endocrine part, okay, so this endocrine part of the pancreas, what is that known as? This is known as islets of Langerhans, right? So this islets of Langerhans, this have some cell that is called as the beta cells, right? So this beta cell will produce the insulin, but whenever beta cell produce the insulin, this is not actually produced in the insulin state, it is actually produced in a inactivated state. So what is that inactivated insulin produced by the beta cell called? This is known as the proinsulin. Okay, what is the structure of the proinsulin? Proinsulin is going to have three chains. First of all, three chain. One is the A chain. Next is the B chain, right? This is the B chain. And the third one, this is the C chain. This is the C chain, right? So this C chain, this is actually one interlinking chain. This C chain will later on will disintegrate, this will disappear and keeps the A and B chains together. So whenever it will be activated, then what happen? This A chain and B chains, they will come closer. This A chain have 21 amino acid and the B chains, they have 30 amino acid. Okay, so remember this insulin, this is the insulin. This insulin have how many amino acid? 51 amino acid. In this A chain, the 7th amino acid of the A chain with the 7th amino acid of the B chain, they develop the disulfide bond, and the 20th amino acid of the A chain binds with the 19th amino acid of the B chain. The second disulfide bond is formed, and this way our insulin is synthesized in our body. Okay, so it is very much difficult that we can able to extract the insulin this way. Okay, so just see what is happening. And yeah, one thing also is very much important, that Sankar gave the molecular structure of insulin, that this insulin have 51 amino acid, who for the first time gave the idea that is it is made up of, it's a polypeptide, it's made up of 51 amino acid, it is going to have A chain and a B chain. This A chain have 21 amino acid and B chain have 30 amino acid, and total this insulin have 51 amino acid. This was given for the first time by Sankar. So remember that. Now just see, this insulin has to be synthesized artificially in the biotechnological lab, because see, human insulin cannot be a good source of the people who are suffering from diabetes, because each and every individual will produce only that amount of insulin they need. Now if it is collected from a different species, then allergic reaction can occur. So that's why we have a technology how actually this insulin is synthesized, right? So we know, suppose this is the A chain, this is the B chain, right? So A and B chains, what are they? They are the protein, right? So these proteins, they are formed from what? They are formed from a specific mRNA, isn't that? So this mRNA was synthesized from whom? From the gene fragment, from the DNA segments, isn't that? So this is the A polypeptide producing DNA, and this is the B polypeptide producing DNA. Okay, now this is the process of transcription by what mRNA is synthesized, and then this mRNA undergo translation and produce the A and B polypeptide. Now just see what is happening. The scientists, what the scientists have done? The scientists have collected the mRNA. Okay, they have collected the mRNA by northern blotting technique. Okay, there is a blotting technique. I have already mentioned in the earlier chapter that there are different techniques we have for the separation of mRNA, that is the northern blotting. mRNA, it was they are isolated. Okay, mRNA by northern blotting was isolated. Okay, so this is the mRNA for the A polypeptide producing gene, and this is this. Okay, this is B polypeptide producing mRNA. Now after that, they are allowed to undergo reverse transcription. So after reverse transcription, it is going to produce the DNA, that is actually one complementary DNA. Okay, this is the cDNA. So this complementary DNA was collected. So this cDNA, whenever it was collected, it was introduced separately in separate E. coli. So for that, we have collected the gene fragment, we have isolated the gene fragment, then separately into the vector. Okay, into the vector, the cDNA for the A producing polypeptide and B polypeptide producing the cDNA was constructed into a vector and made a rDNA. Are you getting me? So this rDNA was constructed separately. It will be introduced into the E. coli. So whenever inside the E. coli they start to express, it produce the protein, right? So it was introduced into separate E. coli. Inside the E. coli it starts to express. Okay, so this A cDNA, this A polypeptide producing cDNA and B polypeptide producing cDNA was inserted inside the E. coli, and both of them they start to express. Express means it produce its own protein, that is A and B polypeptide, it synthesized. So after that we have, by downstreaming process, we isolate the A polypeptide and B polypeptide. Okay, we have found the A and B polypeptide. They're allowed to mix in certain condition, and after that we can able to get the disulfide bond in between the 7th, 20 and 7/19th amino acid of A and B chains, and for the first time this insulin, which is known as humulin, this humulin was constructed artificially. So the company Eli Lilly, for the first time, remember that Eli Lilly constructed the first insulin, genetically engineered insulin, which is known as humulin, was constructed, and later on two another companies, that is Eli Lilly and Ranbaxy. Both these two pharmaceutical companies, they developed one analog of human insulin that was known as humolog. Okay, and after that this humolog is now been used to treat the diabetic patients. So this way actually the whole biotechnology, how it is helpful to produce the artificial insulin, not to have any allergic reaction, and also can properly work. This is the whole idea behind the production of the humolog. Okay, remember that this is all about the insulin. In the next video we will see the other discoveries or other uses of biotechnology in the field of same medicine. So that's all about the production of insulin. You need to know the structure of this proinsulin, insulin, and how it is produced. This is going to be very important part. In boards also, in NEET also, the questions are asked in many different ways. So yeah, that's all about the use of biotechnology in the field of medicine. The first one, that is genetically engineered insulin. So I hope you understand. Thank you.